So this is a panel on high data rate instruments. One of our panelists has already introduced himself, Stephan, so we're going to skip you for a minute. And I just would like to invite each of the remaining panelists to just introduce themselves and really just say why you came here, what was of interest to you, what challenges do you and your company face, and what you hope to impart to the audience here and what you hope to get out of it. Okay.
Great.
Please go ahead.
Sergei Sorkin, I'm the Director of Marketing and Information Technology at 3I, or Intelligent Imaging Innovations. Unlike my colleagues here, we don't do EM, but we do, the company is actually founded on software. There's a couple of postdocs in '95 who decided that it was too difficult to set up commonly needed experiments on their microscopes because there were so many devices that needed to be coordinated for each capture. And so they created this software that we still use today, slidebook software. It's not the same as '95. And the company has evolved since then from a software company to a dealer for microscopes, and from there to a manufacturer of optical, electronic devices and microscopes themselves, along with a number of partners across the board. Carl Zeiss, one of our closest partners, DDN, one of our more recent partners, Microevolution, something that we also provide in our software, one of our partners. And I guess we cover a large net of fluorescence microscopy products, starting with spinning disk confocal, multiphoton turf wrap, laser ablation, and ending up most recently with light sheet technology. So we are a sub-license of an exclusive license to sell lattice light sheet. We have somewhere like 21 installations of lattice light sheet around the world. So we took a fairly difficult instrument and we delivered it to many top researchers around the world. And we have a collaborative style of listening to their problems, suggesting an instrument, and then working with them to the end of each project, and being very flexible, custom, and providing technology to fit that experiment. And the reason I'm here is our partnership with DDN, and storage is obviously the bedrock of all these instruments. You have to store it somewhere, and so we are keenly interested in this field and delivering good, flexible, and accurate, and consistent technologies to our customers.
Hi everyone, good afternoon. My name is Pakzad. I'm here from Gatan. The headquarters is in California. I'm based out of California. So I think most people, I'm not sure actually. So we are a company that make accessories for electron microscopes. So we make cameras, spectrometers, holders, different equipment that are attached to electron microscopes. I am the camera specialist, so we have we would like to say the latest versions of our cameras are the fastest cameras. So we generate quite a lot of data. My background is not life science, I'm a material scientist, but we also generate quite a lot of data because we like to look at materials live in the microscope, so we collect videos as we are pulling or pushing or biasing a sample. So our latest camera, the K3IS camera, generates maybe 2 or 3 multi-terabyte data per hour. And so that's one of the reasons I'm here. We collaborate with DDN, and so we need ways to store the data and then be able to process it either offline or as we are collecting the data to give feedback to the microscope and change the experimental parameters. And I'm very happy to be here. I'm learning quite a lot of what other people are doing in the field, and we like to learn that and apply it to material science and the tools that we provide for all the EM community.
Hi, I'm Jeff Lengyel. I'm from Thermo Fisher Scientific. excuse me. I'm the principal scientist for electron microscopy life sciences for all of the Americas. And so Thermo Fisher, we are the leading provider of electron microscopes and cryo-EM. So 2 years ago, there was a company called FEI I used to work for that we were acquired by Thermo, so that no longer exists. Some people still use this terminology. But you'll actually see these microscopes at the end of the day that there's 3 at Rockefeller, so 2 Titan Cryos and Thales Arctica. And I should mention, these instruments produce a lot of data, at least a terabyte or multiple terabytes per day. And that's going to grow in size, particularly as there's more multimodal and correlative imaging. So data storage and data processing is a huge issue in this field, and it's constantly evolving. So I'm really here just to hear about your concerns or needs. So I mean, on our side, we don't provide data storage, but this is a critical issue because it's so linked to production of the instrument. And so, bottlenecks are quite critical, so how can we overcome them? So thanks for the invite, and I'm happy to contribute.
Great. Okay. Thank you all. So I wanted to start the discussion, as any good philosophical work does, with a little bit of definitions. I'm glad to see some of you got quantitative. We're supposed to be talking about high data rate instruments. So what is high data rate, or what do you consider a high data rate to be? We heard that. In many of the talks, a terabyte a day or half a terabyte a day is something that may not necessarily be high anymore, I guess, in the context of this room. And I think there's also another issue there, which is the time scale, right? We can talk about bytes per day, but we also have concerns about how quickly that data comes. Is it going to be a terabyte over 20 minutes or over a few minutes, and then we don't generate any other data for the rest of the day? Or is it sort of this, I won't say slow, but I'll say inexorable flood of data that just never stops and keeps coming at a constant rate? I think each of those things has different challenges, and I'd just be curious to hear how you characterize your instruments or the problems that you work on across those different timescales.
I can go.
Yeah, sure.
The latest camera that I was just talking about, the K3IS camera, so we have about 24 megapixels, and the image rate is about 75 frames per second. So about less than 2 gigabytes per second. So that's why I was saying about a couple of terabytes per hour. And that's the frame rate that is coming raw as digital micrograph files from the camera, and we store those as they're coming off of the camera onto the disk. Now this disk could be either a local disk on the PC that is running the camera and the digital micrograph, or you can have it directly connected to another storage like the DDM storage and store the data directly on to the server where you can do offline processing. So that's the maximum data rate that we're doing now. Other IS cameras that we have, or when they are being used for CryoEM, where you take a lot of images, but they're still images, the data rates are slower than this one. We spend quite a lot of time when we developed the K2 and then now the K3 just to be able to handle the frame rates that are coming off of the camera. And the rates are defined by 2 things. One, how fast we can read out the sensor, so how fast the camera can generate the data. But we are also a lot of times limited by how fast the data can be written onto the computer or the server. And we are kind of, with the camera, getting to the point where the computer is the bottleneck. So we would like to be able to write the data, maybe have parallel writing the data, so that we can keep up with the camera. So that's the detector side.
Yeah. So I can comment on that. So to give you some perspective, our Titan Cryos, many of them are collecting 1,000 images per day. Now it's not a static image, it's actually a stack of frames. So we actually collect movies, no longer are you doing a single shot. And this could be a 30, 40 second acquisition at 40 frames per second. And to give you some perspective, NYSPC used to collect 1,000 frames or 1,000 movies per day. They're now collecting around 4,000. So it's quite intensive. When we developed our own camera called the Falcon 3, we actually connected it to a 70 terabyte scratch drive just for data transfer, because this can be rate limiting, because actually the transfer was one of the biggest issues. And so we put in our connection, we can transfer a 1 minute acquisition at 40 frames per second in 3 seconds. to give you some time, because we just wanted to mitigate that lag issue. But data transfer is one of the bigger issues, because it's not only do you have to transfer to local storage or long-term storage, a lot of people are actually now transferring data internationally, and this is becoming a big issue. How do you do it through cloud computing or other infrastructures? Some groups are actually still putting data storage on external hard drives and shipping them places in FedEx, which is just terrible. The worst thing you can ever imagine. But yes, usually a lot of people are collecting for 3 days continually. So 12,000 movie stacks, which can be 15 or 20 terabytes.
We're on one mic now. Yeah, from our end, I guess the most common and most greedy sensor in our world is the SCMOS 2K by 2K. It's kind of the baseline for data capture. It's about a gig a second with overhead. And the trick to that is it's all, and there was a little bit of a talk about this earlier, with microscopy, it's always a trade-off. And so one of our instruments, the lattice light sheet, is an instrument that can image almost infinitely without damaging this live organism that it's looking at, live cells or even small embryos, things like that. And so for us, it's more temporal in terms of total file size. So we'll do anywhere from 500 megabytes a second to 2 gigabytes a second. But we have to sustain that over a very long period of time, very consistently. So before, researchers would do experiments and phototoxicity would kill the specimen in, I don't know, a minute, 2 minutes. So they would have long intervals between each capture in order to provide less light to the specimen and not damage it as much. And now they're imaging at a smaller frame, 512 by 512, at like 1,000 frames a second, taking many, many, many Z stacks, all at very fast rates, and that yields really large total file sizes. So I don't know, for us that's a terabyte to many multiple terabytes. And frankly, our constraint is the same. It's really the compute technology. It's how many postdocs you can afford to sift through all that technology, and more recently, machine learning and AI on how to process that data without costly resources. So.
Well, in case of the Multisim, we do roughly 50 terabyte a day max speed. The thing is, it's not really clear in its active research how to get from the image data to the information you need. Therefore, or you really want, and therefore everybody wants to store everything, and let's face it, these are multi-million-dollar instruments. Nobody is willing that the computer is slowing you down anymore. So that means, let's put it that way, if a microscope is 5 million, you should at least put maybe 1 or 2 million of it in computer resources there in order not to slow it down. and I think this is my feeling, what the customers also, maybe 20% or so is maybe willing to spend on it, at least in the first place. And then the question is, how do you then on the long run, how long do you store that? Okay, where does that go? Is that then afterwards on the cloud, and how often do you access it? Do you pay for that every time you need it? Other thing is, are there funding agencies willing to pay for it? I think that would be one of the next steps. Is there the NIH coming up then with a grant where you say, okay, you can access 10,000 times this and then do your science with it? And all these things need to be sorted out, and I don't think they are sorted out yet. And it's also about publishing rights, who's the first author, who's the last one, and so on and so forth. There are many things to discuss there. It's getting philosophical again.
It's like, well, I don't think it is. Sure.
Do you want to my colleague here?
So introduce yourself.
Yeah, Kyle Crosby, also Carl Zeiss, Multisim team with Stephan. Maybe you can comment, I think you alluded to it a little bit, but about the parallel nature of our architecture and how we do acquisition currently with basically off-the-shelf computational no high performance computation, but maybe just give them some idea of how it's designed right now and the scalable nature of that and where we think we might have gaps still.
Yeah, well, we use simple pizza boxes, right? In the end, you have an NVIDIA grabber, a card in the end, and for each channel, and this is basic. This is really standard off the shelf. You attach some glass fibers and then you record the signal. The thing is, this is not really sophisticated in a sense that we put some intelligence in there. You could think about much more. You could think about getting a feedback loop into the data acquisition. This is what you really want to have, It's like, is the image good enough for what I want to achieve? This is the question. Is there information in the image? If you can decide on that, you can either throw it away, take a new one, or improve the image, or just skip it. These are all things where a lot of these, let's say, edge computing could really help a lot. And I think also where what you want to do is really put a cluster next to it, a mini cluster, let's say. Get rid of the first 10 major artifacts and then really boil it down to what you really need to do further downstream processing.
I should say that this is really meant to be an open discussion, so anyone in the audience that wants to chime in, you don't have to be sitting at the front of the room to either ask nor answer questions. So I think what we've heard here is that there's a danger that the limiting factor to progress will be just the ability to either transit or ingest data at the rates that these instruments are capable of producing them. I'd like to poll the audience and ask what people who use these instruments or run facilities that serve other people are finding their current constraints to be. Is it time on the instrument? Raise your hand if your constraint is time on an instrument. If you're running a facility that's booked at 100% and that's your constraint, or is it where you can put the data? Okay, so you're limited by that. Where are you from? NYSBC. Okay, so I'm just curious if a few other people want to be part of a statistical sample of what their constraints are in this field.
Increasing analyst time.
Analyst time. So it's the knowledge worker analysis at the end that's holding it up. Okay, right. Okay, quiet audience, but okay. All right. So I think it's a little interesting that we're being warned that this tsunami is coming, but it seems that on the other side of the room, maybe we're looking at the wave approaching the beach, but we haven't gotten wet yet. Okay, interesting. One of the constraints that I think we face here at Rockefeller is actually instrument time. So far, we've been lucky enough to be able to handle all the data that's coming off of our CryoEM and other high throughput platforms. And those platforms are booked 90% of the time, which makes our resource center directors happy in terms of capacity utilization and so on. And we feel that we're limited by not or wasting some of that time with samples that are not the best, or bad settings on the instruments or so on. So we have a profound profound interest in real-time quality assessment, quality control, and so on. And I think that does lead into doing as much processing upfront as possible in real time. Stephan, I think your talk referred to that, and some others as well. So that's something that I think really can't happen without the deep cooperation of those who make the instruments and the software that drives them. So I was wondering if you could elaborate a bit further on what each of you are doing to make that possible, both with your own solutions and also enabling others to tap into data at the earliest stages, analyze it, and feed back to the way the instrument is run.
Oh, sorry, I guess it was turned off. Yeah, so we're really in a semiconductor model of how do we maximize the uptime and throughput of the instrument. And it's not just about number of images per day, how do we ensure that the quality is uniform throughout the data acquisition? So one of our main initiatives was actually rewriting our whole system architecture and our alignments to streamline them, and actually the microscope will now auto-diagnose its own alignments and say, is the instrument in the proper situation to collect data? Because you don't want a user that tweaks some setting and your acquisition is no longer perfect. So we've done that process. We're doing a lot of sort of auto alignments and how do you actually, ensure that you're getting the best quality data. And then we're putting a lot of effort in saying, how do you pick the proper sections to acquire? So how do you look at optimal ice thickness and other parameters? So these are front end. A long-term vision is, yes, we want to be more proactive, is also doing on-the-fly sort of reconstructions to do initial quality assessment, to say, stop acquiring if your data is not high enough quality and go to these things. That's our long-term vision. We're not there. This is also tied in intimately with sample preparation. We have a big initiative, and we're also querying academics to see what's going to happen if there's some breakthrough in that in the commercial process. Yeah, but we're intimately aware of the issues that you're suffering. And if you look at the instruments in the US, they're generally quite overbooked. So many have 2 or 3 month wait time. So it's a huge issue. So you might only have 2 days every 3 months on an instrument. So you want to make sure you have the maximum use of this. Also in that same vein, a lot of our initiatives are how do we also generate lower end screening tools, so you can do a lot of your low end screening analysis at a much cheaper instrument, and then make sure that your grid is perfect when it goes into the cryos. And then we can also put connectivity and also picking regions in a low end instrument, send those coordinates eventually to the cryos, and it automatically collects data in an analogous model to the synchrotron. So if that helps.
So I agree that throughput is very, very important. So making high quality images are important. So bigger, faster sensors, having a good microscope, combination of a good microscope and a good sensor, having a good software that is reliable to do the work for you without having a grad student sitting there and watching it for 24 hours. Or a couple of days, that's also important. So making everything automized. So these are all, you completely covered all the cryo side. But so if I go to the material science side, when we do in situ microscopy, so we spend quite a lot of time preparing the right sample system and conditions to look at the structure of the sample that we are interested in. And the same thing, having time on high-end microscopes is expensive and it's hard to get. So you prepare your sample, you think about your experiment, and then you want to run it. And you don't want to just collect data, you want to be smart and collect the right data. So what we did a couple weeks ago actually on microscope was what I kind of pointed to, is that as you're collecting the data, you can actually run scripts to access the data that's been stored maybe a few frames ago and process that. For example, look at the crystalline structure and see how that's changing in time, or see, for example, if you're melting a sample, is it now molten or not, and then give that information to the camera or to the microscope so that it can change the parameters, maybe increase the temperature, reduce the temperature, and have that live feedback. And being able to do that, you need to have the computing power. You also need to be able to control and talk with all of these equipments at the same time. So having access at the data that's been captured and then having control on the equipment that's running the experiment is also something that I think is very important, and that's where people are going. So adding this to the experiments, I think that's the next level. You don't want to just be sitting there and collecting terabytes of data and then later on having to go over that data for weeks and trying to get what you needed, and then maybe you find out after you process all of that terabytes that, oh, I need to repeat the experiment because this is really not what I needed to get for my paper. So I think that's where we are going next.
So from the end of our bread and butter being live cell imaging, many of our customers are focused on HPC and just doing as much as they can in real time. From our end, we focus on acquisition, reliable acquisition, and a solid platform in our software, Slidebook, to do bidirectional interoperability with other tools. So we've worked with researchers to develop, for example, some live cell tracking. So as you imagine, you're taking a capture of a live cell, and it's moving. So it may be out of focus, it may be out of the field of view. And in order to deal with that, this one particular case, we developed a hook into MATLAB for a customer that he already had developed an algorithm. He was happy with it, and we were able to feed the data to MATLAB, bring it back in, adjust the capture parameters, continue the experiment. And this is all very quick in real time. On other types of workflows, it may be, it may not be MATLAB, it may be something else. It might be custom made code, and for that we have an API that hooks into our proprietary file format, which is it's a binary file format. We have a robust API that lets you just hook in whatever programming you're doing, and we find that in our field there tends to be more computer scientists now working on these problems with the researchers, and we prefer to stay out of the way and just facilitate as we're asked, and that's been a really good model for us so far.
So we have this research partner program where we try to learn as much as we can from the customer. We have also an open API there to access the Multisim, at least vast majority of the functions there. Interestingly, nobody used it so far. Everybody wanted to have it. No new functions came up. Other thing is, like, we have an open file system. We just write out bitmaps, text files, very basic, very simple. We found that customers work very well with that, on the other hand. I think the improvement is always coming if you approach it on a really broad basis, starting with the sample prep to all the downstream there, which is not so easy, because you need to be an expert in all of that. However, I learned that once you have then such a high throughput instrument, everybody wants to use it. Even the PhD student where the sample is not ideal. I think you can greatly improve throughput in just getting a second instrument, which is maybe not so costly, and check the sample before you put it in. And then have, when the sample is ready and you have also clearly defined your project, set it up on a high throughput machine, and then get the results. However, what's then coming out at a certain point, the projects get much bigger. Whenever you have a faster machine, right, the next double click is like, I need to have 1,000 times more data. So I think this is just in our nature. Always want to have more.
So I have a question, and this ties back to what, George Vasek, moderator, ties back to what Jason asked somebody earlier, but it also ties in with what some of you talked about in real-time analysis, and that's thinking about the raw data. What needs to be kept, for how long, what can you get to a point where you're only keeping process data because you're maybe processing it all on the instrument. You bring it, the computing to the edge, and you just give them a final answer. I don't know what that is, but is that foreseeable, or is it the case that you can't do that and they do have to keep the raw data for reanalysis or other purposes, as Jason alluded to?
Very different for different customers. Some of them appreciate a good EM image. Some of them don't even want to look at the images anymore. They just want the information coming out. They, I think, want to understand what's happening there, a clear referencing of how you measure it and why you did it and why it looks like that and not different. But once you have that, then it's fine. I think then you don't need to have the raw data. You can live with some processing of the data. Very different from customer to customer. Physicists tend to always want to look at the raw data. Biologists less, I would say.
What he said, but I think we kind of tend to operate on a worst case basis, and that was part of the reason why Slidebook uses a proprietary file format. This was an issue with keeping many files and organizing those files over the the years, and there have been a lot of solutions invented since then for better organization of your files. But our file format allows you to store kind of all of the experimental captures through the process, the raw data, the deconvolved data, de-skewed in some cases data, et cetera, analyzed all in one place. So it just makes it a little bit easier to adapt to whatever the particular researcher needs.
What they said. So I talk from my experience. First of all, before collecting the data, so for many years, people who do take videos of what is happening in the microscope and do in situ microscopy, because the samples are precious, they're hard to make, they would just start recording in the beginning of the session, and they would just collect data for hours and waiting for some reaction to happen. And so you would maybe have 1 hour of just looking at the sample and nothing is happening. So a few years ago, we made a tool where you don't really have to start recording data until you see something happen. And I think that's also important. You don't need to fill up your disk. That's not how you publish papers, but you need to fill it up with real good data. So that tool is called a look back, and you just basically have a buffer of the data being stored in some memory for you. And once you see the reaction starts, you can go back in time and store that part of the data and add it to your data set. So I think that's one tool when you're recording the data. We also, for data analysis, provide tools where you can mine the data. And I think from what I see from I agree it depends on customers, but a lot of our customers collect the data and then because you have to have a large field of view, because, for example, if you're heating particles, you don't know where they're going to go. But once you have the data, we have tools to crop the image in time, in space, and go and mine the data and just keep the part of the field of view that you're mostly interested in and reduce the data size and then apply more analysis to that to learn more about your structure. That's basically the trend that I'm seeing in the material science side.
So in the CryoEM field, actually Jason introduced this perfectly. The issue is it's not that advanced in the image processing realm. And so a lot of people will try to do a reductive approach. So as I mentioned, you don't acquire a static image. You get a series of frames and you process this, you pre-alignment. Some people only save those pre-aligned stacks, but the whole programs to do that alignment are improving constantly. And so you're getting better things out of it. So my belief is you should always keep all of your raw data. And so even sometimes you process data and it fails for some other reason, it's just because the algorithm is failing. It doesn't mean the data is actually bad. And with improvement of algorithms, you can actually reuse data you thought was unusable. So this also ties in with the problem of how do you do self-assessment, because the algorithms are not great enough to really say, is this the best data or not? So you can put certain checks in and say, do I have particles? And you can make a certain assessment, but you can't do the whole pipeline because the pipeline's evolving so much, which makes it quite problematic. And so, but many labs will actually do some processing, delete their initial data sets, and do reductionist things, but it's always problematic, in my opinion, the state we're in.
So if we do evolve to the point where many of these steps become, I guess, first agreed upon and then incorporated into the instruments, which in many of the cutting edge fields we're looking at here may be a year or 2 or 3 away, do you think we see peak data egress and this is really a transient problem in this space? Or do you think that as we solve that problem, we'll just output more information and just soak up all the bandwidth and disk space that we'll ever be able to muster?
Yeah, so, okay, there's a couple different points here. So first of all, the interesting thing is theoretically we should use a very small fraction of our data. We're actually collecting way more than we need based on physics, and that's because of problems in the sample, issues in data image processing, misalignments in microscopes. A lot of these are actually soluble, so and throughput of the instruments should improve. So, but it's hard to predict finally where we will go. Now, the funny thing in the CryoEM field, the algorithms are really not that complex. The issue is actually signal-to-noise ratio, and that's the real fundamental problem. So you're overcoming just poor signal-to-noise, and it's through averaging. So that's always going to be a rate-limiting step, and the fact that biological samples are sensitive to the electron beam. But there's things we can improve. Now the question is, how far are we getting close to that theoretical limit? Because we're not going to go to the optical limit of the microscope. For many samples, it's not going to make any improvement. It's just the intrinsic sample problem. But I do think within time, many of these steps should become standardized. In the X-ray crystallography community, they have a very standard file format, they have very specific programs. CryoEM should go to that point. I mean, theoretically, there's no reason it can't. It's just more a fact that the technology has been evolving so fast that the image processing has not been keeping up with it. Now, image processing is starting to come up with or deal with like all the new advances in technology and resolution that we presented. I don't know if that helps.
I'm not 100% sure where we're going. I think everybody wants, again, I'm going to talk about cameras. Everyone wants larger field of view. Everyone wants bigger sensors, high resolution, faster, so they get higher temporal resolution. You can see things move and change. But I think we also need to, like we were talking about, implement these feedbacks into what we are doing during the experiments and time on the microscope. A lot of times, well, TEM samples to begin with are really, really small, and sometimes people just look at 1 or 2 or 3 samples and they want to expand that to a microscopic behavior of the material. So I think if we implement that feedback and we can collect really just the data that we need, we reduce the time on the microscope and we can look at, instead of 3 samples, at 30 samples. And then that would be a lot better statistical information of what you really have in the material. But yeah, it's hard to say. Really, it's exciting times, but it's hard to predict what everybody, it's good to get feedback from you guys of what you really want and what the challenges are. And usually that's what we try to take back and then think about how we can solve them to make life easier when you run the microscope.
Yes, is the answer to your question. In our experience, just from what we've done, working with basic scientific researchers since 97, I think in the late 90s we were selling a whole rack, like 18U of servers, just to do simple deconvolution. And now microevolution can do it on a single GPU almost in real time. And it was days before. So I think the nature of researchers, I mean, there's computational researchers doing their jobs, there's storage researchers doing their job, and biologists should keep up. They should try to use all the resources they can. That's our take. So keep buying microscopes and keep buying storage and keep buying GPUs and et cetera, et cetera.
Yeah, don't worry, HPC guys, you're not out of business so quickly. It is like variability in biology is huge, and before we can describe that, we can buy some more computers. I totally agree. Bandwidth is an issue, storage is an issue, and especially look at one human brain. If you have one, it's a good start, but you in the end want to look at many of them in health and disease. So that will still take a while until I think researchers are satisfied.
All right, well, we've asked you all to crystal ball that. We'll check back in a few years and see how we did. I think we've only scratched the surface here, and we could go on for quite some time, and I hope we do during the subsequent breaks and reception later tonight. But I think we'll go back to our program of talks for now. So I'll thank the panel, thank the audience, and